All right, everybody, this is going to be our chapter 5 on microbial metabolism. Metabolism is nothing more than the collection of all the biochemical reactions that are happening within a microbe. Ultimately, again, we're talking about reproduction of this organism. So in order for the organism to do that, it must have the appropriate amount of energy and be capable of making that. So these statements are going to be essentially the guide for most of your metabolic processes. And those are that every cell has to acquire nutrients. Again, these nutrients are going to have to be used to build things within the cell. These are also going to require energy from light, or they're going to take the energy from the breakdown of nutrients. Again, we cannot get energy from nowhere. It cannot be created or destroyed. It has to come from somewhere. In this case, either the bonds of the things that we eat or from light. Energy is stored in adenosine triphosphate, ATP. This is going to be where most of the currency of that energy is going to come from. Cells catabolize nutrients to form precursor metabolites. Of course, we need building blocks, and those building blocks are essential for building cells. And so we'll talk about those. And these metabolites, ATP and enzymes, are required for anabolic reactions. And we'll talk about how these enzymes facilitate those processes as well. Enzymes plus ATP can form macromolecules. Of course, they're used to add things like phosphate to these molecules and other components, and we'll talk about that. Cells have to grow by assembling macromolecules. We'll talk about the pathways as we get a little bit closer to the end of this chapter, and that they produced only when they have doubled in size. Again, you cannot just create a cell with nothing. You have to be able to create everything that those cells require to be functional once they completely separate. And so to start off, we have the two different pathways. We have the catabolic pathways and we have the anabolic pathways. The catabolic pathways, these are typically the breaking down of the molecules that are large into smaller ones. This is an exergonic reaction, typically followed with an increase of entropy. And then you have the anabolic pathways. These are the building pathways, the synthesizing pathways, and these are androgonic. Typically, they require more energy that they're going to release. And so here we have a cell visualizing both catabolism and anabolism. Of course, nutrients are going to go into the cell. Then those nutrients are going to be used to either use the energy to create new things like ATP, or they're going to be dissipated or lost as heat. And then some of that energy is going to be used for the building processes, the anabolic processes, where they're forming larger compounds, larger molecules, and then those are going to be redirected either to cellular structures, processes, or storage of longer-term energy like carbohydrates and lipids. Now, before we get into more complex things, we have to understand some of the basics, and those basics involve redox reactions or oxidation reduction reactions. And what these reactions revolve around is the transfer and the acceptance of electrons. So one molecule is going to take an electron or accept an electron. The donor molecule has to give it up. And so these reactions always occur simultaneously. They don't happen like very, very far apart. They typically happen pretty close to each other as well. We are going to be dealing with three important carriers, and these are NAD+, NADP+, and FAD. So these have different structures, but they do a very similar thing, and that is to carry or to accept electrons. And so here, going back to that concept, so for something to be oxidized, it has to lose an electron. So here we see an electron donor molecule that has the electron in red, and that is giving it up. And so that is known as oxidation, when you've lost that electron. Now reduction is when the electron that was given to us by the donor is accepted. So the molecule accepting the electron, that is the reducing agent, that is the reductive part. And so again, this is known as a redox reaction. Whoever loses the electron is oxidized. Whoever accepts the electron is reduced. One of the statements that we looked at earlier said that ATP is going to be the form of energy most of these cells are going to use, and that is correct. So in order for us to make ATP, we first have to transfer the energy from somewhere, and that somewhere is from the nutrients that cells absorb. So this or these bonds that are broken are going to take that energy that is released and they're going to connect an ADP, an adenosine diphosphate, and they're going to add an inorganic phosphorus to it, leading to the formation of ATP, which is an unstable high energy molecule. So this is the process known as phosphorylation. And so you have several different types of phosphorylation. You have substrate level phosphorylation, which can happen without the use of ATP synthase. Typically something like this would happen in glycolysis. Then you have oxidative phosphorylation, which is the process of forming ATP with the presence of oxygen. And we'll talk about the electron transport chain as one of the mechanisms that makes this possible. And then we have photophosphorylation, which happens in the presence of light. Of course, any anabolic pathway for the most part is going to use ATP as a mechanism to synthesize them. Again, ATP is going to be feeding these molecules or these enzymes to transfer that energy into larger molecules. And enzymes are a very, very important part of this. Enzymes are going to increase the likelihood of a reaction by decreasing the activation energy or the amount of energy required to start a chemical reaction. And this is mainly due to the mechanism of their action. They're either going to force things together or force things apart. Typically, they're going to be your builders or breakers. And you have several different categories of these. You have hydrolases, isomerases, ligases, also known as polymerases, lyases, oxidoreductases, and transferases. And so typically you will see ASE at the end of an enzyme. So you have your hydrolases. These are catabolic in nature, typically used to break down things like fats. You have your isomerases, which are neither anabolic nor catabolic. Their only function is to rearrange or reorganize. Ligases or polymerases are gluers, so they are anabolic. They create large molecules. Lyases, in this case, they can be catabolic. Whether they are splitting things using water, they're super important for breaking down some of those bigger things like proteins. Oxidoreductases, these are the ones that are transferring electrons from one molecule to another. And then transferases, these are the ones that can modify a functional group by moving it to something else. And these may be anabolic in nature. Make sure you're familiar with table 5.1 as it includes all 6 of them. So enzymes are really important. They can create these complex reactions and they will activate based on different things in their environment. So you have things like apoenzymes, which are inactive until you essentially create a functional larger enzyme by the cofactors that are present. So these could be ions, it could be other enzymes. And once they bind to their cofactors, they produce a hollow enzyme, which is now almost like a molecular machine that's a little bit more complete. And things like this could be things like a ribozyme, for example. So here you have the holoenzyme, which is the apoenzyme that now has been activated by the coenzyme and an inorganic factor. And all of these, when they're combined, they create the active site. And now this active site is ready to accept molecules and either build them or break them. So here we have a list of important cofactors of enzymes. So please make sure you take time to examine table 5.2. So here we have a chart just of the amount of energy required for a chemical reaction to start. As you can see, in the presence of enzymes, it requires a lot less energy when they are present. Here we have the basic enzyme substrate complex. So on the left, you see the enzyme. On the right, you see the substrate. So what happens when the enzyme and the substrate combine is that there is a temporary change in the conformation of the enzyme. So the enzyme essentially changes its shape in order to facilitate the chemical reaction. This creates the induced fit of the enzyme to its substrate, forming the enzyme substrate complex. So here we have an example of an enzymatic reaction. You have the substrate fructose 1,6-biphosphate or bisphosphate, and here you can see that molecule going into the enzyme in step number 1. In step number 2, there's a conformational change and the enzyme substrate complex is formed. Once that complex is formed, then the activity begins. In this case, this molecule is cleaved and you separate it into 2 parts. So this is one of the initial parts of glycolysis. After this process occurs, then the halves of this molecule will be released from the enzyme and the enzyme will be ready to bind another molecule. So in metabolism, these are going to build or break things. And just like you saw earlier, in that case, that molecule was broken. But there are several different things that can influence whether or not that enzyme is going to be functional. These are things like temperature, pH, enzyme and substrate concentrations, as well as inhibitors. And so here we see how there is an optimal temperature to different enzymes. Of course, every enzyme is different. In our case, this would be something that would look like us. So as you can see, most of the enzyme activity is at its highest, right around 37 degrees Celsius, which is approximately what normal human body temperature is. Then we also have the denaturation of proteins, and this can happen because of different things. Temperature can break the sensitive hydrogen bonds. You can also have things like the changes in pH, for example. So some bacteria, for example, can increase the or withstand greater changes in pH as to not destroy their the bridges that are formed within this. And so whenever that protein becomes denatured, it unfolds. If it does not have the correct shape, it loses its function, and so it can no longer work as intended. We also have the substrate concentration saturation point. Let's say that you have a big factory and you build bicycles. Let's say that you have 50 workers and you have a large shipment of 100,000 bicycles. At a time, there can only be 50 bicycles being made because you only have 50 workers. Doesn't matter how much more material you bring. You could bring in 1 million more bicycles. You're only going to be able to process 50 of these bicycles at a time. And that's the concept that we see here at that saturation point. It doesn't matter how much substrate you add. At some point, you're only going to have a limited number of enzymes that are actually working at the same time. Now, controlling enzymatic activity is a very difficult thing, mainly because you either have to turn processes on or off, and sometimes they might not work as intended. There are things that can interfere with these processes, and so cells have different mechanisms to deal with this. You have some enzymes that have activators that the enzyme will only work in the presence of these activators, and you also have inhibitors, so the enzyme will not work in the presence of an inhibitor. So here we have an example of an allosteric activator. So this enzyme right here is non-functional. It does not have the correct shape to bind to its substrate. However, when in the presence of the allosteric activator, it changes the conformation of that protein, allowing it to bind to a substrate, form the enzyme substrate complex, and then have the capacity to do work on that substrate. Now, there are also, like activators, inhibitors. So there could be some that are competitive, some that are non-competitive, and some of them that are controlled by a feedback inhibition mechanism. So here we have on the top figure, figure A competitive inhibitor. So this one, the competitive inhibitor will bind to the active site of the enzyme, and no matter the concentration of the substrate, that is not going to be removed. So you have the blocking of that active site. In a reversible competitive inhibitor, however, let's say that you bind a competitive inhibitor to that site. If you were to increase the concentration of the substrate, this would be enough to remove that competitive inhibitor. And so by removing it, you're able to restore the normal function to that enzyme. You can also have allosteric inhibition where the inhibitor binds to the active site, binds to a space known as the allosteric site, changing the active site's shape so that the enzyme cannot work properly. A great example of this would be phosphofructokinase, if I'm not mistaken. So in this case, let's say that you're breaking down too much glucose and you're creating a lot of ATP. In this case, the ATP will bind as an allosteric inhibitor, preventing the enzyme from working, preventing the breakdown of more glucose. Again, if you already have enough, the ATP itself will act as an allosteric inhibitor, preventing it from breaking down even more. So here we have a feedback mechanism of inhibition. So the substrate is going to be broken down. Once that substrate breaks down, it's going to keep following the cascade of those enzymes until eventually it reaches to its final intended product. When that product increases in concentration, then the end product is going to act as an allosteric inhibitor by preventing the first enzyme in the cascade from working. So this is a negative feedback where you increase that substrate, and the level is appropriate, then the process will turn off. Now, when it comes to carbohydrates, carbohydrates are one of the primary energy sources for a lot of organisms. That's going to be their primary source of energy. And so glucose is one of the things that a lot of things can metabolize. So fermentation and cellular respiration are two of the most important processes that use a lot of these carbohydrates. So here's kind of the summary or the big picture. The big green block on top, that's going to be glycolysis. We'll talk about glycolysis as a process in itself. This is also known as the EMP process, and we'll talk about that in contrast to the ED process a little bit later through this lecture. But glycolysis is a series of steps that take glucose, phosphorylated, and then split it down the middle to create two pyruvic acid molecules. Now, these pyruvic acid molecules can either continue through the process of respiration, where they're going to go to acetyl-CoA processing or pyruvic processing, which leads to acetyl-CoA, and then those eventually going into the citric acid cycle to remove most of the hydrogens and electrons so that you can feed the electron transport chain. On the other hand, if pyruvic acid is there and there is no oxygen present, or it's a type of bacteria that doesn't really process glucose that much, then you have fermentation. And this could lead to two processes, which we'll talk about a little bit later as well. And these are lactic acid fermentation and alcoholic fermentation. But I guess we're going to go through the first respiration side in the first place. So glycolysis is a series of steps in which there is some substrate level phosphorylation and there's a direct transfer of energy, and it can produce a few ATP, and this is very minimal. However, this is not really the potential that a full glucose molecule has. It's 10 steps and it's divided into 3 sub-steps, and these are the investment phase, the lysis phase, and the conserving phase. And so here we have this entire molecule, or the entire molecule's processing all the way to pyruvic acid. So we start with glucose. So glucose is a 6-carbon compound. Now the investment phase is called the investment phase because you have to input ATP into this process. So by introducing ATP, what the first couple of enzymes are doing is they are phosphorylating the glucose molecule. So after the first one, you have glucose 6-phosphate, and then you have fructose 1,6-bisphosphate. And so by doing that, by adding the phosphorus, you're charging up that glucose molecule. And so in the next step, this is where the lysis stage begins. That 6-carbon compound is broken down into 2 3-carbon compounds. One is glyceraldehyde 3-phosphate, and the other one is dihydroacetone phosphate, DHAP. Sorry, the words are really little. And so as these compounds are broken down, they're modified, especially the DHAP. And so ultimately the goal of the lysis phase is to get 2 glyceraldehyde 3-phosphate molecules. Once those molecules are made, then now you start the process of some redox reactions. So you introduce an inorganic phosphorus to these 3-carbon compounds, and with the help of NAD+, you are producing NADH. So that first redox reaction has occurred, and as a consequence, now you have 2 phosphorus or phosphate groups on either side of the molecule. So now during the energy conserving stage, this is kind of your payoff phase. So now ADP comes along, and as ADP comes along, it takes one of the phosphorus or phosphate groups from the glyceraldehyde phosphate, and it creates ATP. And then after that, water is removed from this molecule, and you're left with PEP. And then once PEP is made, another ATP molecule comes in. It removes the second phosphate group, and now you have a second molecule of ATP. So total, this process happens twice. It happens one for every G3P molecule that you have. Therefore, you invested 2 ATP at the beginning of this cycle, or at the beginning of this process, and at the end, you are ending up with 4. And so this right here, this is that substrate level phosphorylation where we're getting minimal amounts of ATP. So net, you have a total of 2 ATP in this process. So here we have that holoenzyme, and this is kind of showing you the process of how PEP gets converted into an ATP molecule and pyruvic acid. As you can see, this enzyme here is splitting the bond between the oxygen and the phosphorus. adding that phosphate group, that inorganic phosphate to ADP, creating ATP, and you're left with that skeleton, which is pyruvic acid. Now, this pyruvic acid still has to be processed further, and there are 3 more stages that need to occur in this step. And this is cellular respiration. This is now something that needs to happen within the mitochondria or within another bacterial cell. Glycolysis can happen in the cytoplasm, just about any cell, but these processes are a little bit different. Okay, so here, now we're going to begin to process pyruvate. So pyruvate has a carboxyl group attached to the end of it, and through the process of decarboxylation, you're going to remove a CO2. So what you're left with is acetate. Now, acetate gets processed further by the addition of coenzyme A, and after that coenzyme A is attached to acetate, you also have another redox reaction occur where you introduce NAD+ and you leave with NADH. Again, these electron carriers are really, really important. I don't think I've mentioned the importance of these, at least here yet. So by removing that additional hydrogen, you're also removing the electrons, and these electrons are going to be extremely important for the electron transport chain. So the breakdown of that glucose molecules involve removing some of those hydrogens, taking the electrons with you and moving them or shuttling them somewhere else. And we'll talk about where that is as we get closer to the ETC. But moving on to this step, here now you have the addition of coenzyme A to the acetate compound. Eventually, at the end of this process, you will form acetyl-CoA for the acetate molecule with the addition of the coenzyme A. Now that acetyl-CoA is formed, you have 2 of those molecules. You have 2 CO2 molecules and 2 molecules of NADH. Again, this happens twice because you have 2 pyruvates at the end of glycolysis. So now we're going to go into the Krebs cycle. So all that acetyl-CoA that is produced, now you can try to maximize the removal of the energy from that little skeleton molecule. So you're going to do that by taking in as much NAD+ as possible and FAD and removing as many electrons and hydrogen atoms as you can. Of course, in prokaryotes, this happens at the cytosol, and it happens in the matrix of the mitochondria in eukaryotic cells. And there are several different reactions that you have. You have the anabolic reactions, isomerization, redox reactions, decarboxylations, some substrate level phosphorylation, and hydration. And all of these molecules are different, or all these processes cause different things within this. But ultimately, what these processes are doing is they're preparing molecules to strip them of those hydrogens and those electrons. Without some of these reactions, it would be almost impossible to draw out or break those bonds, releasing the energy that is held within them. And so this chart has a lot of those reactions. I think your text does a good job at explaining, so I encourage you to read that text. One of the things that this chart fails to mention or visualize here is the addition of double bonds and the addition of some hydrogen groups that you have here. However, the main thing that I would like you to note about this diagram is all of the redox reactions that are occurring here. And so you have several different places at which NAD+ is entering. You also have FAD being converted to FADH2, and you have the formation of some substrate level GTP, which is a chemical compound really similar to ATP. Instead of adenine, however, it's guanine. And so you have those things. You also have hydration. And again, hydration in this case, it's not necessarily for anything other than the addition of those hydrogen atoms so that you're able to remove that in the next step by introducing NAD+ and then taking NADH out of that. And so it's just modification and rebuilding of citrate, which eventually is going to give rise to the next I guess the next electron carriers in this case. So total out of here, you're going to probably take out the equivalent of about 15 ATP molecules. So by the end of this Krebs cycle, you should have 2 molecules of ATP, 2 molecules of FADH2, 6 molecules of NADH, and 4 molecules of CO2, all of which, again, are really important for contributing to the next step, which is the electron transport chain. So this electron transport chain is exactly what its name tells you it is. It is a chain that transports electrons, and electrons are negatively charged subatomic particles. And as you may or may not know, whenever you take an electron away from hydrogen, it becomes a positive ion. And so what hydrogen is going to do is it's going to follow these electrons around. And as these electrons are being followed by those hydrogens, the proteins that are found in this membrane are going to be pumped either into the extracellular space or into the matrix of the mitochondria. And so what happens here in this case is you use those electrons to establish a chemiosmosis process. It's essentially the buildup of hydrogen on one side of this membrane, and once it multiplies in the thousands, then you're able to accept these hydrogens through the, or you're able to release these hydrogens through the tunnel found within ATP synthase. And once that gate allows those hydrogens to pass through, it's kind of like a little hydroelectric plant. As this molecule spins by the exiting of those hydrogen ions, it's going to allow for the formation of ATP. And so in this case, you have to have some sort of final electron acceptor. So after all those electrons are used to kind of lure the hydrogens into that space, it has to be captured by something. In the case of cellular respiration, oxygen is going to be that final electron acceptor. Although it doesn't always have to be oxygen, you could have some things else accepted. However, that final electron acceptor is going to determine how much energy you're actually producing by how strongly it'll bind to those and how good it is at accepting those electrons and removing that free energy so that you don't waste any. So that proton gradient, again, super, super important. As you can see here, the energy that is removed is almost at zero. So you're essentially taking out as much energy as possible from that glucose molecule. So some categories of interesting carrier molecules in the electron transport chain. You have your flavoproteins. So these are going to be your initial acceptors of things like NADH, and they're able to take the molecules and strip them of its functional parts, specifically the hydrogens and the electrons. You then have your ubiquinones, so aka these are your coenzymes Q that are shuttling these electrons from one part of the electron transport chain to the next. Specifically, we're talking about the cytochrome complexes that they're moving in between. These complexes are also capable of pumping hydrogen into the side of the opposing, or the opposing side of the knob on ATP synthase, so that they're building up that proton gradient. And then these protons, of course, they contain iron, which is really, really important for establishing that redox potential. So you're able to take the electrons, transfer them, and of course, use them for whatever you need. In this case, for pumping that hydrogen into that space. So at the end of this process, in this case, oxygen is going to act as your main final electron acceptor. So as hydrogen makes its way across that selective gate on the ATP molecule, it's producing ATP. Oxygen is capturing those electrons, those hydrogen that's going to form water. And so it's interesting how this entire process works, because it's all powered by that series of electron carriers that you've been taking out since the initial phases, right? You have a lot of this built up already. So this process is kind of the culmination of the breakdown of those bonds, the taking of the electrons, the taking of the hydrogens, that little bit of energy, and using it to synthesize ATP. So here we have the visualization of that cycle. Again, the filler proteins are taking those electrons and that hydrogen from NADH. The ubiquinones are shuttling those electrons through that cytochrome B, cytochrome C, A, and A3. And then, of course, right there towards number 3, you see how hydrogen is being pulled out by oxygen, trapping it, and those electrons are following that molecule once they're done being used to shuttle the hydrogens into that space. And as you can see on the far right, you see those hydrogen ions flowing out through that ATP synthase gate, and as they're doing so, they're spinning, allowing the molecule to form, or the molecule of ATP to form. Now, this isn't always easy and functional. Sometimes you can have things interfere with the process. Things like cyanide in this case could actually attach to that last part of your cytochrome A3, and by doing so, they're preventing from that the redox reaction from occurring. So you are preventing that from reaching the oxidative state again, so that the process can resume. And so in this case, it'd be really similar to the effects that oxygen deprivation would have by preventing the process from continuing, that redox fluctuation between reduction and oxidation. So that chemiosmosis, like I mentioned earlier, it's an electrochemical gradient. It's there because it's able to generate ATP. And those protons are going to follow their electrochemical gradient. Again, they're in high concentration on the outside. As that gate allows them to go through, they're going towards where there is less hydrogen, following the gradient. This, again, is known as oxidative phosphorylation because it requires the process of oxidation in order for the electron transport chain to function properly, thus making about 34 ATP molecules. From a single glucose molecule, by the end of this process, you will have about 38 total. So this chart just shows you the ideal activity of prokaryotic organisms during respiration and how they're able to process one of those glucose molecules. Now, there is diversity when it comes to carbohydrate catabolism. Not every organism breaks down the same. Some pathogenic bacteria use the ED pathway in contrast to the AMP pathway, which is the one that we just covered. Again, this is only in prokaryotes. So instead of producing two, they only produce one ATP, one NADH, one NADPH, in contrast to the two of the traditional pathway. You can also have the pentose pathway, which is an alternative to glycolysis. Of course, it's less efficient because it only produces very, very few molecules, including things like NADPH. I mean, NADPH is really, really important. It's one of the precursor metabolites for nucleotides, steroids, fatty acids. However, again, in terms of energy production, is not as good as the EMP pathway. Fermentation, of course, is when you cannot have, or you do not have oxygen present to be able to accept some of those carriers, and so eventually the carriers end up returning those electrons and hydrogens to one of the ending molecules that are produced, eventually going back and freeing up that redox potential for them to continue the process. And so you have the partial oxidation of these sugars. Again, highly, highly inefficient process. But if you're a microorganism, this just might work. So here we have those two pathways. You have the lactic acid fermentation and you have the alcoholic fermentation. As you can see, in this case, the NADHs that are produced in that initial process are going to be returned to the molecule, in this case, producing these byproducts of alcohol and lactic acid. This table compares aerobic respiration, anaerobic respiration, and fermentation in terms of their potential for ATP production. So make sure that you do examine these. Now, these different processes that are produced as the glucose molecule is only partially produced can lead to different types of acids or different types of products. It can have different types of acids, different types of alcohols produced, and they each kind of correspond to different microbes. So each one of these microbes creates a different product. And again, they're very, very diverse. They can produce a lot of different byproducts. And so this is one of the things that industrial microbiology is really important. Now, other catabolic pathways that are worthy of mentioning that we should also look at that are covered in your text are the different conversions of these precursor metabolites before they can be something useful again. So here we have the catabolism of triglycerides. So triglycerides are a glycerol molecule and some fatty acids. So after this molecule is broken down, the fatty acids are released, and the glycerol molecule itself, by the addition of ATP, can be converted into DHAP, which is one of the first halves that is produced through glycolysis. So after this molecule can go into that same pathway, be converted into eventually glyceraldehyde 3-phosphate. The fatty acids, on the other hand, they undergo a series of reactions that allow coenzyme A and water to modify this molecule, making it shorter, And as it makes it shorter, it's drawing out some of those hydrogens, some of those electrons. So once those are being split, those products can go off into the electron transport chain. On the other hand, as these fatty acids are getting shorter, they're breaking down some of these fragments into fragments of 2 with the addition of the coenzyme A. Eventually, that little byproduct can be shuttled into the citric acid cycle. And so different components of these fatty acids can be driven into different processes within the catabolism of glucose. And so different products can go into different phases. The same can be true for proteins. So when you have a polypeptide, those polypeptides can be broken down via hydrolysis, and then once they're processed into individual amino acids, they can go through the process of deamination, where you remove the amine group, and then those byproducts end up going into the citric acid cycle, as they're really, really important for the building up of citrate. Now, when it comes to photosynthesis, these organisms are capable of taking CO2 and water and creating some of these complex carbohydrates. Now, it's really important to note that the electron transport chain that is in cellular respiration and the electron transport chain in photosynthesis have some major differences. Of course, one of them is driven by light. The other one is driven by the energy of the bonds within the glucose molecule. So in the case of photosynthesis, it's just important to realize that there's something different here. It's not working off the energy of a molecule, it's working off the energy of sunlight. And so CO2 and H2O take some interesting pathways here in order to be able to synthesize some of these molecules. In this case, you have chlorophylls, which are pigments that are capturing light, and then you will have some similarities when it comes to the cytochrome complexes in the electron transport chain. However, they're slightly modified for different processes. And of course, you can utilize different wavelengths of light. You're dealing with 680 and 700 nanometers typically. Now, in terms of their organization, you have the thylakoids, which are the sacs, and then you have the stacks of these called the grana, and the stroma is the space in between them. A lot of these chlorophyll molecules are going to be kind of embedded within these membranes, and because they have an increased surface area, you're able to increase how much light is actually being captured. In prokaryotes, this is happening within the cytoplasmic membrane. In eukaryotes, it's happening inside the inner membrane of the chloroplast. And so here you can kind of see the organization of these pigments, but also how they're embedded within the membranes, and then how those membranes are folded kind of to maximize the surface area for photosynthesis. There are 2 photosystems involved in photosynthesis. You have photosystem 1 and photosystem 2. Photosystem 2 is going to be the initial one, and photosystem 1 comes after that. Typically, photosystem 1 can recharge the electrons once they've lost their energy, and photosystem 1 is the one that initially charges them. However, there's something interesting about photosystem 2 that doesn't happen in photosystem 1. When we talk about electrons and the electron transport chain here, we don't have that traditional pathway where you're having the breakdown of a glucose molecule to give you some of those NADH compounds, FADH2. These electrons are going to come from somewhere else, and that is where water comes in. So during photosystem 2, what happens is, as these electrons typically going through what's known as non-cyclic electron flow, they could be lost, they could be dissipated, they could be captured by another element or atom and molecule, and eventually they will have to be replaced. And so water is what restores the electrons to the electron transport chain. If you have no water and you lose those electrons, you cannot continue the process. Now, photosystem 1, it can recycle some of those, but it doesn't have the capability to break them off from water. And so photosystem 2 is kind of important as it can do that, right? So here we have kind of the initial part of the photosystems. So light energy is going to hit chlorophyll. This chlorophyll is going to excite the electrons, and the electrons themselves are not moving yet. So all the light is doing right now at the moment is exciting the electrons, and that energy is moving from one chlorophyll molecule to the next. So this is known as resonance energy transfer. And this energy is transferred from molecule to molecule until eventually it's going to have the capability to jump. And that is not going to happen until it reaches the reaction center. So at the reaction center, you're going to have an electron acceptor. And this is kind of where the same thing that was happening in cellular respiration, or the ETC for respiration, now is happening here. These electrons are being captured. The electrons are being trapped by a shuttle. And then that shuttle is taking them across the electron transport chain. As it's moving them through the electron transport chain, this is what's building up the proton gradient. This is what's concentrating the hydrogen, because hydrogen is the only thing that can spin ATP synthase. If you can't spin the ATP synthase knob, you cannot make ATP. And so this process is essential for the functioning of the electron transport chain. So again, here we have some of those same structures here. Again, they're not identical. But as you can see in that initial step of non-cyclic electron flow on the bottom, you see water molecules coming in, you see the hydrogens going out, you see the electrons being stripped and replaced as the ones that are in the electron transport chain are captured by other molecules. And at the end of the process, you have that ATP being formed. As you can see, there are also two photosystems. These photosystems are connected. They're connected by the complexes that are found within the membrane, not necessarily connected directly, but indirectly by moving or shuttling electrons from one of these cycles or one of these systems to the next. Table 5.5 has a comparison of all the different types of phosphorylation. I encourage you to look at that. Now, the light-dependent or independent reactions are what happens after you have a lot of that hydrogen being built up. In this case, the ATP and NADPH that are being made in that cycle, the photosystem 1 and photosystem 2 dependent cycle, this is now going to go into the process of carbon fixation in the Calvin cycle, or the Calvin-Benson cycle. There are 3 steps to this. First, you have fixation, then you have reduction, and then you have regeneration. And so here we have that pathway, just so you can examine it. And so here, for the first time, you start having the introduction of CO2. A lot of people think that CO2 just removes the carbon and then the carbon goes in and makes the glucose, but the oxygen is actually coming from splitting of the water during photosystem 2. Here, CO2 is coming in as a gas. That CO2 is starting to combine with the molecule ribulose bisphosphate, so also short, we're going to call it RuBP. So CO2 is a 1 carbon compound, RuBP is a 5 carbon compound. So as they do that, they combine into 3 carbon compounds known as 3-phosphoglyceric acid. And then as that molecule continues through that cycle, ATP in this case is going to be added. That ATP is going to phosphorylate the molecule into 1,3-biphosphoglyceric acid. After that acid is made, then NADPH is going to come in. It's going to give the hydrogens and the electrons. It's going to also remove some of those phosphates. And after doing so, you're left with G3P. So something interesting happens to most of this G3P. 5 of these G3P molecules, even though this produces 6, are going to go back and regenerate ribulose bisphosphate, or RuBP. The one that isn't, that is going to be used to synthesize glucose. And so that G3P is going to combine with another G3P as the second spin occurs, and then you will generate finally glucose 6-phosphate. After that, the reverse is happening as you had in glycolysis, and a glucose molecule will be formed. So one of these per spin is going to be used to make glucose, the other 5 to regenerate RuBP. Now, other anabolic pathways include reactions that can proceed in either direction. Those are known as amphibolic pathways. And so you can have ATP coming from a lot of these anabolic reactions. You can have ATP being released from some of these reactions, but they're all interesting nonetheless. There are 12 precursor metabolites that are required, that are very, very functional, and that are going to generate different compounds within the breakdown or the building of glucose. And so it's important to look at some of those pathways as well. So table 5.6 has a collection of them, the pathway that generates the metabolite and what they're used for. So here we have first the biosynthesis of complex carbohydrates. So from the Calvin-Benson cycle, you have CO2 entering, you have all that stuff occurring. Some of those, once they're made into G3P, can go straight into the fructose 1,6-bisphosphate production. You could also have glycerols from fats, after we mentioned how they were broken down, going into that same pathway. You can have those then eventually go into fructose 6-phosphate, which can be developed into peptidoglycan, which is an important component of some bacterial cell walls. Others can be made into glucose 6-phosphate, which can then turn into glycogen for some storage. And then some of that can turn into glucose, which will eventually lead to starch and cellulose. Now, let's say that you don't have some of these. Let's say that all you have are fatty acids. Well, those fatty acids can also synthesize into acetyl-CoA. Acetyl-CoA can reverse into pyruvic acid. And then from that pyruvic acid and the addition of ATP, you can have oxalic acid and then finally G3P. And this process can continue. So regardless of what you are feeding into this cycle, that's one of the things that is wonderful about cell's machinery, is that you can go into gluconeogenesis from either the fats or glycerols or fatty acids. You can essentially build them from there. You can also even take amino acids from the proteins that you deaminate and actually bring them back into the G3P production. And so there's a lot of different ways that you can build glucose molecules. And so the same is true for the synthesis of triglycerides, fatty acids, and lipids. You can have that glyceraldehyde 3-phosphate converted into DHAP by reversing the reaction, turn it back into glycerol, and then synthesize some fats, triglycerides. And then you could also take some of the acetyl-CoA molecules that are made during glycolysis, and through a reverse beta oxidation process, which is discussed in your text, you can take that, turn it into fatty acids, and then synthesize the other molecules that are necessary. This includes phospholipids, triglycerides, and fats. And so all these can be used at different stages to create the products that are needed by the cell. Now here we have what happens, or what can happen, to amino acids. You can have the addition of an amine group to oxaloacetic acid to create aspartic acid, and then that aspartic acid can be used at different stages to build different compounds. And then you also can have the oxaloacetic acid from that citric acid cycle be converted into a aspartic acid or alpha ketogluteric acid, which eventually are going to be part of the citric acid cycle. And then some of the products of that cycle can go back into some of the others and feed into those systems as well. And then when it comes to nucleotides, the process is really, really similar as you see with most of the other ones. As long as you have some of those precursors, they can eventually go into the different pathways. Here, through the process of glycolysis, you can have a glucose 6-phosphate or have that coming directly from photosynthesis. And if this microorganism goes into the pentose pathway or pentose phosphate pathway, you can create ribulose 5-phosphate, and then from there create pyrimidines or purines, and with the help of things like PABA, can be converted into folic acid, or the aspartic acid from the citric acid cycle can be used to add to those nucleotides, eventually synthesizing DNA and RNA. So whatever is coming into the cell, whatever nutrients are available, they can always be converted and modified into different stages of these metabolic pathways. And based on what the cell needs, those will be created based on the materials that you have. And so they're super, super interesting, the way that they could just be created and be synthesized in different ways. Make sure that you examine some of these in the reading. Now, the regulation and integration of these metabolic functions, they all have to do with what is available. So typically cells are going to be catabolizing the more energy efficient choices. So sometimes they'll break down things that are easier to break down than others. They're also going to create metabolites based on the needs of the cell. Whatever is available is also going to be used. So cells are going to take what they have and they're going to use it to build what they need. Eukaryotic cells are a little bit different. They can isolate these enzymes in different pathways within the organelles, and that's kind of one of the benefits of having organelles, is you can isolate, separate, and compartmentalize reactions. Some cells can use allosteric sites to inhibit the specific enzymes just to prevent them from going further. For example, if you don't need to break down glucose further, that ATP is going to be enough to prevent that from happening. You have some of these feedback mechanisms that are slowing down pathways, that are speeding up pathways. It really depends. When you have too much of something, however, typically you're going to slow or stop these processes to prevent the breakdown. For example, if we kept on breaking down glucose, without that regulation, we would have to be eating every second of the day to keep up with the amount of energy that's being broken down. Now, there are different regulatory mechanisms. Here we'll talk about two, the gene expression and the metabolic expression. So you can have the timing and the amount of enzymes being produced controlled by genes, and some of these could be triggered by the environment. You could also control that expression by controlling the activity of proteins or enzymes. So this is by releasing inhibitors that are preventing these enzymes from working. Here we have the integration of all of these different pathways. Make sure that you examine this and that you look at where these processes can feed into the others and what things they have in common so that you're able to understand some of the similarities between them, some of the differences, and what the final product of these are. Again, the intended pathways for most of these metabolic processes is going to be the synthesis of ATP and the breakdown of glucose. But again, you can always go back to wherever the cell needs there to be more products as the cell is in need of them.